Following on from our prior lesson on mitosis, today we are going to have a look at meiosis. Now, meiosis is another form of cell division with the main purpose of producing gametes. When we talk about gametes, our examples are the sperm and ova. Sperm and ova are the sex cells which originate from, in males, the testes, and in females, the ovaries. These gametes are required in order for the process of fertilization to take place. Fertilization requires that each of our gametes has a total of 23 chromosomes, or what we could refer to as a haploid number. Meiosis as a process results in daughter cells with half those number of chromosomes, so our haploid number, and in these gametes, in our final daughter cell, only one chromosome from each of the original homologous pairs is present. So as a quick recap from our prior lessons, when we see the number and letter 2n, this refers to a diploid number, which in humans refers to 46 chromosomes. When we see that n, we are referring to haploid, meaning 23 individual chromosomes. When we look at our phases for meiosis, I'm just going to single out, again, interphase here. Interphase leads into meiosis. Interphase is not strictly a stage of meiosis itself, in the same way that interphase leads into mitosis. So when we're talking about interphase, we're going to refer to this as a stage of cell division. And the stages that we looked at yesterday were our G1 stage, our synthesis stage, and our G2 stage. Within our interphase, it's important that we note that during synthesis, this is when our DNA is replicated. Now, fortunately for us, when we look at meiosis, our stages repeat. We're not learning anything majorly new over mitosis, but we are learning some different things to help us distinguish between these two stages. So as a quick overview, meiosis is made up of two divisions. In our first division, we have our separation of homologous pairs, which leaves us with two daughter cells, each with a total of 23 chromosomes. Now, per chromosome, we have two sister chromatids. This is different from our second division, where our final product is one chromatid per chromosome. During our second division, we have separation of those original identical sister chromatids, and instead it leaves us with four daughter cells, each with 23 chromosomes. Here is a very quick overview looking at that arrangement and how it moves throughout the meiosis process itself. So starting off, we have our one diploid parent cell. Our homologous chromosomes are replicated, so we end up with a copy of, say, our blue chromosome and a copy of our green chromosome. And then we get to this first meiotic division. And this is where our homologous pair of replicated chromosomes separate. This leaves us with our second division, where we finally get our sister chromatids separating as well. So while we start in the same way as mitosis with a diploid number, we finish with a haploid number in each of these chromosomes. Here is another diagram showing each of the stages that we're about to unpack here today. So skipping over interphase, which we had a look at in the mitosis lesson, but we will start today having a look at the stage of prophase 1, followed by metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. This will then be followed by a restart, moving into the second division where we have prophase 2, metaphase 2, anaphase 2, telophase 2, and lastly, cytokinesis 2. Looking at prophase 1 now. During prophase 1, the most important thing for us to note is that the nuclear membrane breaks down. The nuclear membrane breaks down, and during this time as well, our centrioles move towards the poles of our cells and they begin to make that spindle. We can see that spindle on our diagram here. Our homologous chromosomes become visible. They were once our chromatin, but now we're beginning to see them as our chromosomes. And our key point of distinction today, something which makes prophase 1 different to prophase in mitosis, is that crossing over occurs. And to put a long story short, our crossing over is a form of a process known as variation. Variation will be one of the upcoming lectures, so we're not going to dive into it too much now, but we will touch on it on this next slide. When we talk about crossing over, we already know that our homologous chromosomes are paired up in prophase 1. What we don't know, however, what we haven't discussed, is that they exchange small pieces of genetic material with other chromosomes. Now, we can see this quite clearly in our diagram here, where we have this crossing over between our orange and our blue chromatids. As we have a look, we see that this part known as the chiasma is where these two chromatids cross over. What this crossing over does is it produces new variants. It produces new combinations of genes, and as a consequence of that, we can increase the variation that we see in our 4 gametes. This is a longer diagram from the textbook breaking down this process of crossing over, and we will come back to touch on this in our variation lesson. Moving along down to metaphase 1, so after our homologous chromosome pairs have appeared, after the process of crossing over has potentially occurred, we move into metaphase 1. And in metaphase, as we know from mitosis, our chromosomes line up along the midline or the equator of the cell. They also, at this stage, attach to the spindle via their centromere. If we're having a look at our centromere, it is this dot that I'm drawing in now in yellow in the center of each of our homologous chromosome pairs. These are our centromeres. On the other hand, using my blue pen here, if I circle these features right at the end, they are our centrioles. And our centrioles, again, are what produce that spindle. As we move into anaphase, similar to mitosis yet again, our homologous chromosome pairs separate. It leaves us with one chromosome moving to each pole. And we can see that we have our chromosomes are non-identical here. We have some unique variation, again, as a process of crossing over. Now, these homologous chromosome pairs are separated by those spindle fibers which contract, and this means that our sister chromatids still remain attached to one another, but they move towards the poles. As we now look to telophase 1 and cytokinesis, during telophase, we note that the nuclear membrane reforms as our chromosomes gather at those poles. Our end result after cytokinesis being that pinching of our cell membrane, we end up with 2 unidentical haploid daughter cells. So to break it down, again, 2, nice and easy, unidentical, so we've had some variation. When we talk about haploid, we know that they are the N number. N equals 23 chromosomes, and they are daughter cells. From telophase 1 and cytokinesis 1, we then jump straight into prophase 2. Prophase 2, the key thing that we're looking for here, as done previously, is we're looking for those centrioles moving towards the pole of the cell and beginning to make that spindle. Now, prophase 2 is occurring in both of our daughter cells that finished at the end of the first meiotic division. Moving into metaphase, our chromosomes again line up along the equator of the cell and they attach to the spindle at our centromere. Anaphase is where we start to look a little bit different to where we were in anaphase 1. During anaphase 2, our sister chromatids are now pulled apart, and they're pulled apart again by the same structure, those spindle fibers. This means that one chromatid from each chromosome is going to travel towards those opposite poles of the cell. And we can see that really clearly in both of our cells here. Each individual chromatid being pulled towards opposite sides of the cell, no longer attached to its sister chromatid. As we jump down into telophase 2 and cytokinesis, we see again in telophase our nuclear membrane forms around each set of chromosomes, that spindle fiber disappears, and lastly in cytokinesis we get that division of cytoplasm. Our end result now is slightly different to our first meiotic division. The key difference being the number of cells produced. We have 1, 2, 3, 4 genetically different, genetically unique or varied haploid daughter cells. So to overview of this lesson and the topic of meiosis, gametes are our sex cells. They are our way of producing new offspring. The sperm cell will one day meet the egg cell and it produces a unique organism with 46 chromosomes. This 46 chromosomes, again, is our diploid number, and it comes from a haploid sperm cell and a haploid egg cell.